Hello, I'm Taylor Kuebler in collaboration with Paul Buscemi and Dr. Yuk Sham. And for today's BICB Symposium, I'm going to briefly review a couple projects I've worked on more recently with modeling and simulation of copolymers and how they can sometimes increase a lipid bilayer's resistance to mechanical rupture. And I'll also look at some simulation of micelles and how they can interact and fuse with other lipid aggregates and small molecules. So previous research has shown that unstable membranes have biological consequences. One such consequence is Duchenne muscular dystrophy, or DMD, and it's a fatal disease, and it's characterized by progressively worsening deterioration of striated muscle. Previous work has shown that copolymers, such as poloxamer or P188, can actually stabilize muscle cell membranes, and they can increase these bilayers' resistance to mechanical rupture. Multiple block copolymers have been studied and shown to have varying influence on membrane stability and integrity. In the figure to the right, it's basically showing the pressure that is required to rupture a membrane, comparing both just the base membrane as well as that membrane with the copolymer P188 included. The figure is showing that the copolymer P188, when included with the membrane, is increasing the pressure that's required to rupture that membrane in comparison to the base membrane alone. So with the aim of helping to design copolymers that improve membrane integrity, I've been working to model physiologically representative bilayers and simulate how copolymers influence membrane integrity. So as I said, copolymers can strengthen lipid bilayers by inserting themselves in between the lipids of a membrane and conferring resistance to rupture. In silico, this mechanical stress that leads to bilayer rupture can be mimicked by applying lateral surface tension to the membrane. In the figure below, to the left, it's showing a membrane with no tension being applied, no mechanical stress. And above it is a copolymer P188 molecule with the hydrophilic portions of the molecule in the core and the hydrophobic portions of the molecule forming a shell on the outside. On the right side of the figure, it's showing a membrane with surface tension being applied. And you can see the membrane has been compressed and stretched, and the copolymer P188 has actually opened its hydrophobic core and allowed it to integrate in between the lipids of the bilayer. First, I wanted to model and simulate lipid bilayers with a single lipid type, in this case POP-C, in the presence of copolymer P188 to see if I could replicate results that were reached experimentally in previous work. In these simulations, I observed that how the POP-C P188 system behaved was similar to what was found experimentally. At higher levels of surface tension, as the lipids of a bilayer are stretched from one another, the P188 copolymer actively fuses with the bilayer, potentially improving membrane stability as reflected by a higher level of lateral tension being required to rupture the membrane. But both experimentally and in silico, I found that a majority of current research focuses on lipid bilayers that are comprised of a single lipid type only. For example, POP-C. And this isn't physiologically representative of membranes that are found in nature. So I wanted to model bilayers that were more representative of, say, a sarcolemma cell membrane, which, when compromised, drive the progressively worsening muscle deterioration that's characteristic of Duchenne muscular dystrophy. I wanted to model bilayers with many different lipid types and with different proportions of lipid types and compare how the different copolymers affect membrane stability in these bilayers. Now on slide 6, I built models of bilayers that are composed of POP-C, POP-E, DMPC, DPPC lipids, cholesterol, sphingomyelin, etc. And the video to the right is showing one of these mixed membranes being ruptured. In this case, POP-C, DPPC, and cholesterol make up a bilayer in a 2:2:1 ratio. These simulations help to determine how varying the lipid types and the proportions of these lipids in a bilayer can influence membrane stability, as well as helping to observe how the lipids interact within that bilayer. For example, the figures on the bottom right are showing how lipids of the same type, in this case cholesterol, can coalesce and form lipid rafts, which can lead to local aberrations in the membrane structure, such as increased thickness. Going forward, I'm wanting to look at how these mixed lipid membranes react to increases in surface tension and how copolymers like P188 can influence stability. The next work I'm reviewing starts on slide 8 and is looking at micelles, which are spherical lipid aggregates that are essential to many biological processes. These lipid aggregates, they can be composed of single lipid bilayers, as seen in micelles, or double lipid bilayers that are seen in liposomes and vesicles. Micelles, liposomes, and vesicles are essential to biological processes. They're found in cell membranes, and they're utilized for drug delivery systems. In my modeling and simulation of micelles, I wanted to study what is the nature of initial micelle formation, and how do these micelles interact with other lipid aggregates like other micelles, and how do these micelles interact with molecules like copolymers or drugs. On slide 9, the video on the top right is showing a simulation of 3 identical POP-E micelles fusing and ultimately forming a larger aggregate or a larger micelle. The simulation video on the bottom right shows how the copolymer molecule P188 passively fuses with a POP-C micelle, ultimately forming a micelle copolymer complex. Going forward, I'm continuing to model and simulate increasingly complex bilayers in micelles that are physiologically representative to try to observe and expand upon what has been seen experimentally.